Hello and welcome to the GZERO World podcast. This is where you can find extended conversations from my show on public television. I'm Ian Bremmer, and today I am taking stock of a war that has been 50 years in the making. President Richard Nixon signed the National Cancer Act of 1971, officially launching what would come to be known as America's War on Cancer. Since then, hundreds of billions of dollars have gone into research, prevention, and treatment, and yet cancer continues to kill. Roughly 1 in 2 men and 1 in 3 women in the United States will be diagnosed during their lifetimes, and it remains the nation's second leading cause of death after heart disease, claiming nearly 1,700 lives every single day. But there's been progress too. Survival rates have increased dramatically. Nearly 19 million Americans today are cancer survivors. That's 1 in 18 people alive, compared with 1 in 40 back in 1991. Since then, the overall cancer death rate has dropped by 34%, sparing more than 4.5 million lives. Cancer is personal. Both my parents died from it. If you haven't faced it yourself, you know someone who has. But my guest today gives me reason to believe that technology may be finally tipping the scales in our favor. As artificial intelligence continues to advance, we could be at the beginning of the most significant medical advancement of our lifetimes. Joining me to talk about how AI is reshaping medicine and the fight against cancer is physician and best-selling author of The Emperor of All Maladies, Siddhartha Mukherjee. Let's get to it.
The GZERO World podcast is brought to you by our lead sponsor, Prologis. Prologis helps businesses across the globe scale their supply chains with an expansive portfolio of logistics real estate and the only end-to-end solutions platform addressing the critical initiatives of global logistics today. Learn more at prologis.com.
Siddhartha Mukherjee, welcome back to GZERO World.
Thank you so much.
I've been doing a lot of shows recently on artificial intelligence and how it seems to be affecting so many fields. But medicine comes up a lot, and you exist to a degree at the intersection. Talk about what is surprising to you about where AI is changing the trajectory of medicine.
So just to take a step back first, about a year and a half ago, I created Manus, or Manus AI, in collaboration with Reid Hoffman. And Manus AI is a company that really sits at the intersection of medicine, in particular cancer medicine, and AI. And the mission statement of Manus AI is to create an AI-native pharmaceutical company, which is an end-to-end company. Sometimes you will say for a medicine, cradle to medicine company, which is trying to develop medicines using AI and cures for diseases like cancer using AI. So that's the background of where I'm coming from. So now back to your question. Your question is, how is AI transforming medicine and cancer medicine? And I like to think about it in terms of 3 to 4 very broad silos. And I'll talk a little bit about where we are in those silos and where we aren't in those silos. So the first is the most obvious silo, which is the patient-facing AI silo. It usually involves applications that patients can use usually on their devices, such as scheduling, simple triage, simple questions like, should I have X, Y, or Z? Do I need to see someone immediately, or can I wait? Involves billing, the movement of electronic medical records, the central records, to your device, and potentially beginning to start scraping some of that information for the patient's use. So they can ask, like they ask a chatbot, you can ask your EMR a question, which is, something like, when's the last time I had my cholesterol checked?
So we're not talking about aggregation yet. We're just talking about one-on-one. This is your experience with medicine as an individual patient.
That's correct.
AI is improving.
That's correct.
Yes.
So that field is very full of actors, of people who are doing this. It is, I would say, low-lying fruit AI. It's very helpful to patients. But from the AI standpoint, or from the cures for diseases standpoint, of course, has no real relevance. Then we come to the second category, or the second big silo, which is doctor or physician-facing AI. Now we're talking a little bit more complicated things. We're talking about not just the medical record and how you interface with your physician, but potentially projects that have to do with things like diagnostics. So just to give you a couple of examples, diagnostics that rely on images, visual diagnostics, radiology, so CT scans, MRIs, etc., dermatology, so looking at a rash, looking at a lesion, is it melanoma, is it not melanoma, and finally pathology. Is this breast cancer aggressive or non-aggressive? What do we know about it? What do we understand about it?
And is it true that AI is already more effective than doctors would be in some of these areas? I've certainly seen studies that have implied that.
So the part of the answer is it depends on who the doctor is. Doctors with years and years and years of experience generally tend to outperform what is currently some of these AI systems, whereas obviously a young doctor who's getting experience is not going to outperform these AI systems. The good news about that is that going forward, it's almost certain that, at least to me, it's almost certain that it's going to be a mix of the physician and the machine, the learning machine, and a mix that is going to be, over time, refined, because, as you know, the first day you start reading a CAT scan is, that's your first CAT scan. The AI machine, the machine has usually, it starts with 500,000. So it's going to be hard to compete against those kinds. So just continue down that road.
Yep.
So diagnostics, especially in these image intensive fields. Then comes real data mining and data querying. So your EMR, now aggregated across multiple medical records, electronic medical records, creates a huge corpus of knowledge. That huge corpus of knowledge is untapped for the most part, partly because it's a very complex knowledge. In some cases, like with the Veterans Affairs, in many cases, the knowledge is not in fields. In fact, it's free text knowledge.
It's not on a spreadsheet that's being shared in a broader environment.
And it's not, I would say more importantly, the structure of the data, the underlying structure of the data is not divided up appropriately. So ironically, this unfielded data is actually going to get a move forward. It moves faster with AI because many chatbots are able to read it faster than they could read fielded data. So that's very interesting, because you could query aggregated data and ask potentially very interesting questions like, tell me whether or not, or what kinds of patients had a response to this drug. Are there some characteristics that I can draw out of that? This is now, we're moving into territory that is really interesting, data mining, and this is truly complex data. And I would say that the first applications in this realm in AI are beginning to happen. So that's the second, I would say, broad silo. The third broad silo, which probably is, to me, the most interesting, is true discovery, generative discovery. And there, we're essentially trying to find out that if you have a target, whatever it might be, some protein that is not working normally in a cancer cell, or a protein that's working abnormally in an autoimmune disease, can one develop a medicine that will change the activity of that protein? Now, this is truly interesting to me, of course. In other words, we are trying to create a new pharmacopoeia of chemicals, potentially of chemicals that have never been created before in human history, to address these very complex diseases. Allied to that is, once we have those proteins, what is the best clinical trial that one can run so that you can get the maximum benefit from this drug.
Is this similar to what we saw with COVID vaccines supported by AI?
So COVID vaccines were really, to be honest, not really supported by AI, in the sense that COVID vaccines drew from a history, goes back about 40 years. And COVID vaccines used a relatively simple code, which was the mRNA code. So I would not use COVID vaccines as an example of what AI can do. There are just barely, and I really think just barely, the first medicines that are truly developed by AI coming out. There's a couple of antibiotics that have come out that are quite transformational. Transformational in the sense that their chemical structures, the chemistry of those antibiotics, has never been seen by humans before.
And so how was that developed?
So broadly speaking, there are 2 or 3 ways that one develops these medicines. And it's super important to understand them, because this is the magic promise. First of all, you have to understand that every medicine works by binding to something else. And by binding, it means that it goes and physically attaches itself with, and that attachment has some physical characteristics or biophysical characteristics, which are based on chemistry and physics.
Which will strengthen it, destroy it, change its behavior.
For instance, yeah. Exactly. So once we know that, we can ask a couple of questions. One is, then, if you know the target that you're trying to attack, you can essentially figure out where there's a pocket in that target. So proteins fold in real space, right? So they create folds, and those folds have nubs and pockets and attachment sites of various sorts. That's a natural consequence of the chemistry of the protein. Now, when you identify a pocket, it's like puzzle solving. So just like in a puzzle, you have a little hole, and then you have to put a puzzle piece in. How do you put a puzzle piece in? 2 very broad, it's going to be very broad, but 2 very broad ways that AI can help in the puzzle fitting algorithm. So again, think about how a child solves a puzzle. One way a child solves a puzzle is by, let's say a puzzle it's almost done, there's one piece that's missing, that's that pocket that you want to target. And there are 1 billion pieces sitting on the side. So what a child does is, generally speaking, she starts fitting the puzzle, and then she tries several times, and then realizes that the puzzle has the missing piece has a certain set of characteristics. It has to have a divot on top and a little hole in the bottom, et cetera, et cetera. That's being learned. That's a learned property. And that's why AI comes in, is very important. So then she says, okay, I've learned that property. I've extracted that property as a feature. And now I'm going to go back into the many pieces. I'm going to search for that property. I'm going to eliminate 99% of the pieces that don't have the property. I'm going to search for that property. And now I'm going to fit again. And so now I have a much better fit, but I'm still not there yet. So then I learned that, in fact, the ones that are left have most of the properties, but the one thing that's missing is the little hole on top. So I refit, and so forth. So that's one way that you can create a new medicine.
And the interesting thing, of course, is it's not that this is something that humans couldn't do, given enough time.
Absolutely.
But what AI is doing is creating maximum efficiency, given the extraordinary number of permutations you're talking about.
Yeah.
Okay.
And also remember that those 1 billion pieces that I talked about, they've also been generated computationally.
Right.
So humans could do it. They would first have to make the 1 billion pieces, and then they would have to find the fit. And absolutely, it's what humans can do. And in fact, that's how we've been making medicines for decades. And then there's the most interesting one, the one that we do, and the most interesting variant of all of this. You could have a particularly...
you do it, Manas.
In Manas, yes.
Yes, okay.
We do both, actually, I should say. You could have a particularly rascal child who says, you know what, I don't want to play this stupid game. What I'm going to do is I'm going to take a piece of paper, and I'm going to cut that piece of paper and make it fit into that puzzle piece. I'm going to put that puzzle piece in. Now, what does that mean in real life? It means that you grow the puzzle piece inside the whole. So you learn the rules, and then you begin to take that puzzle piece and say, I understand the rules of that missing piece. I'm going to create, using some chemical rules, I'm going to start creating candidates. I'm going to forget about the billion pieces. I'm going to start creating candidates that will fit into this puzzle piece.
Which may have never existed before.
May have never existed before. This is true generative chemistry. It is true generative AI and true generative chemistry. And some of those pieces, when a real chemist looks at those pieces, human, looks at those pieces, would say, that just doesn't make any sense.
You mean it doesn't exist? I
can't
identify that thing.
So I can't identify that thing. That's okay. That is not a problem. They would say it couldn't exist because I can't synthesize it. I can't make that piece because the physics of it, the chemistry of it, just won't be made. So you discard those. But by the way, when you discard them, you're telling the AI, the machine,
these are things you shouldn't be looking at to try to create the piece, right?
Exactly.
So once again, you're going through this filtering process of, okay, now I know the kinds of things I could do to generate the piece.
Exactly.
Right.
So what you're doing and so the next time around, it won't generate that piece. It'll generate another version of it. And again, you could say, looks okay, but that end of it doesn't really look great. The machine will say, okay, I got it. I get you. Next time I make a piece, I'm not going to put that end in. So what I'm trying to emphasize is the scale and power of all of this. In principle, of course, humans could do it. But every time we do this in collaboration with a machine, the machine learns it, and it learns it forever. You don't need to train a new chemist, a new generation of chemists. The machine will now learn it for eternity. And now you might say, well, what if 10 years from now we realize that
there are ways to create some of these chemicals?
It's not hard. You just tell the machine, go back to it.
Yeah.
And the machine learns in 2 seconds.
Do we have any examples? I mean, again, I know this is coming, but do we have any drugs, any antibodies that have been created with this method so far?
So we have made antibodies with this method. Manus has made some antibodies, very particular kind of drug. And the reason antibodies is actually we just got the results back yesterday. So this is how fast the field moves. Antibodies, because antibodies have antibodies that tend to be stereotypical. And by stereotypical, I mean they have a very particular kind of structure. It's not infinite variation, but a relatively finite variation. So antibodies, the first ones we got yesterday, and we had them for a while, but then we tested them in real life.
And what are they for?
They're for various cancers, blood cancers. So one is for blood cancer called myeloma. There's another one for a blood cancer called a lymphoma.
And why did you decide to start with those?
With those diseases or with those antibodies?
Those diseases.
So it turned out that there were pre-existing antibodies for those diseases. What we trained the machine to do is to evolve them, which is a much simpler problem than starting from complete scratch. And that's the reason that we started with those.
Evolve them to make them what?
To make them bind more tightly, or change the conformation so that they bind in a different place or spot. So we decided to evolve.
And so in principle, a patient with an evolved antibody will have what kind of experience compared to before?
So they will need lower doses of drugs. They might need drugs spaced out more. Most importantly, we hope, and there's good reason to believe, that the toxic side effects of the high doses of the drugs, antibodies, will be mitigated by lower doses of the drugs. Those are the experiences. So potentially more effective, potentially less toxic, and potentially at a more convenient dosing schedule.
The GZERO World podcast is brought to you by our lead sponsor, Prologis. Prologis helps businesses across the globe scale their supply chains with an expansive portfolio of logistics real estate and the only end-to-end solutions platform addressing the critical initiatives of global logistics today. Learn more at prologis.com.
Now, we've been talking a lot about what AI is doing in the field. Now, if I flip it and say, okay, how does one address cancer specifically? Because we're starting into that, and there is the experience that you have with treatment, with identification of tumors earlier on. There's the mitigation of side effects, and of course, there's to what extent you can create a cure. I'd love for you to talk a little bit about what's happening cutting edge in this field right now.
Sure. Again, let's divide up the field into three major categories. As you can see, I'm a big splitter: prevention, early detection, and treatment. Mm-hmm. So what's happening in prevention? Prevention is the hardest. It's also the most impactful, obviously. You want to prevent disease. And almost always in every disease in the history of mankind, there has been the maximum impact has come from prevention. So for a long time, prevention efforts had stalled because prevention really depends on the identification of a cancer-causing agent like cigarette smoke and the mitigation, removal, elimination of that agent, right? But before you can do the elimination, mitigation, et cetera, you have to actually find the agent. And our tests, we had two broad kinds of tests to find cancer-causing agents. So one broad kind of test was a laboratory test, or laboratory tests. So this included putting the chemical onto cells and asking the question, do they start getting mutations? And of course, mutations are what cause cancer. So this was called the Ames test. It was devised in the 1970s by Bruce Ames, and still remains a very important test. The second kind of test, laboratory test, which is very labor intensive, is to expose animals like mice or rats to a suspect chemical and ask the question, do they get cancer? The problem is that we're now finding out that the Ames test, even though it's very valuable, misses a whole host of carcinogens or cancer-causing agents, chemicals. So for instance, I'll give you one example. Asbestos. It's a great example of a chemical which clearly causes cancer, lung cancer, mesotheliomas, and others, doesn't score on the Ames test. Similarly, it's actually quite difficult. You can do it, but it's quite difficult to expose animals to asbestos and get cancers. It's possible to do. The other kind of test is the opposite, is a test which is epidemiological. In other words, you study a population and you ask the question, is there something in the population gets cancer or some part of the population gets cancer? And you ask the question,
what are they exposed to?
What are they exposed to?
Yeah.
Right. So asbestos, of course, comes very clearly in that category. So occupational workers, people who work with, insulation, whereas asbestos clearly have very high rates of lung cancer, mesothelioma, et cetera, et cetera. So, and we've known that for a long time. So there was a discrepancy between the epidemiologists and the laboratory scientists. Right. And it was very hard to sort this discrepancy out because, you don't know what is doing what. A great example of this is that, I don't know if you remember, but at bizarrely, phenomenally high doses, you can get a signal from coffee in some laboratory tests. And so there was a movement for a while in California. You'd have to walk into a Starbucks and it would say,
could cause cancer.
Yeah.
Yeah.
But massive epidemiological studies showed that coffee drinkers have absolutely no change in cancer risk. So again, a weird discrepancy. And these discrepancies kept growing because, and there was a growing sense that we're missing something. So, and what is it that we're missing? So most recently, what's happened is that we've figured out two things. Number one is we figured out that inflammation inflammation defined as a very particular kind of activation of the immunological response, collaborates with chemicals to cause cancer through a mechanism that we're beginning to identify. And so going back to, again, to asbestos, it's very likely that the asbestos particles cause a particular kind of inflammation. We don't know this for asbestos fully yet, but cause a very particular kind of inflammation, which ultimately leads to cancer. Another example of this big recent study, prominent study, air pollution, but a very particular kind of air pollution which is very minute particles of air pollution.
Which we measure.
Which we can measure, of course. Yeah, we measure every day, actually. So these are called PM 2.5 because they're less than, they're less than a particular size, causes cancer. But if you take that chemical and you put it into the Ames test, you don't get cancer. So what's the backstory about all of this? Well, it turns out that these particles get lodged deep in the lung, and they cause an inflammation, this inflammatory response, a very particular, it's not just any inflammatory response. It's not just like having a bacterial infection. It causes a chronic inflammatory response. And that chronic inflammatory response through a mechanism that I won't go into is responsible for cancer. Now, why is that important? It's important because all of a sudden, the universe of things that cause cancer has widened. And now...
And become specified.
Exactly. And now, all of a sudden, really, I would say after years and years of waiting, we have a new theory that it's, cancer is caused by mutations, yes. It's caused by abnormal cell division, yes. But it is also caused by an inflammatory milieu. Now, that leads to a whole host of new potential carcinogens that need to be evaluated. I'll give you a couple of examples. One great example that people are now very concerned about are these forever chemicals. Generally speaking, these inflammatory chemicals tend to be like forever chemicals. So the immune system tries to metabolize them, swallow them up, clean them up. They can't do it. And in response to that, you get cancer, and inflammation and cancer. Now, all of a sudden, there's a new host of things that need to be explored in prevention. So and it's a big deal because we are suddenly finding that we can explore the world, the chemical world and the biological world to some extent, and really begin to think about, is it carcinogenic, is it not, and how to mitigate and remove them, etc. We don't know the answer to, for instance, these forever chemicals, but just to give you, how it changes practice. For instance, I threw away all my Teflon ware because Teflon is a consequence of one of these forever chemicals. So I now cook only on non-Teflon pans. Now,
and would you say, is that proven in your view?
No,
it's not.
No.
So we don't need to tell everybody yet to get rid of Teflon. But you're being abundantly cautious.
I'm being abundantly cautious.
Okay, fine.
Exactly. All right, so then we come to early detection.
Right.
We were using 5 marginally effective mechanisms to detect cancer early. So colonoscopy, pretty effective, unpleasant, but poor compliance. Pap smears for cervical cancer. Mammography for breast cancer. So probably misses a lot of cancers. Low-dose CT for high-risk people such as smokers for lung cancer. Yep. And then the PSA test for prostate cancer, which, as you know, is very complicated. It has a lot of false positives, and it's not really a great test for prostate cancer. So those were the 5. What's happened in the interim is that scientists have invented blood tests that can detect cancer at its very early stages based on DNA that's shed by the cancer cells into the blood. So there are many companies that have grown around this. One prominent one is called GRAIL Therapeutics. There are several others. The problem with these is that they're still undergoing testing. We don't know whether these will ultimately lead to lives saved or lives not saved. There are some very fundamental conundrums why these tests take very long and sometimes still don't yield the right result. I'll just give you two conundrums that are important, sort of thinking experiments. One is the conundrum of false positives and false negatives. So every test has false positives and false negatives. And these tests also have false positives and false negatives. False positive is when you detect a test. When you detect something, it's not, but it's not real. It's not really cancer. False negative is that you say you're fine, you don't have cancer, but actually you do.
You do, yeah.
Yeah. That is one conundrum, but that's not the only conundrum. The other conundrum is actually much more interesting and much more subtle. Imagine two sisters who developed breast cancer at the same moment in time. Let's say they're twins. One of them decides to undergo therapy, and she lives for 5 years. After she undergoes detection, she enrolls in a program, undergoes detection, but unfortunately, and then treatment, et cetera, but unfortunately it doesn't work, and she dies 5 years after. The other sister says, I don't want to do any detection. They don't know they have cancer. But she says, I don't want to do any detection. I want to live my life out as it is. And so she says, I'm not interested. Eventually, the cancer grows and it becomes visible, let's say 4 years from then. And she says, I don't want any treatment. I'm done. And she dies as well. And let's say both the sisters die at the same minute, the same of the day.
Right.
Now, when you're counting, you'll say that the first sister lived 5 years. Her survival was 5 years after the detection of her
But it
cancer.
wasn't actually the case in the sense that we don't think the treatment accomplished anything.
That's right. They both died the same minute in time. So what does that tell you? It tells you you can't use survival
As useful data.
as a useful data.
Right.
You have to use mortality as useful data. Now that creates a problem for a trial for early detection because you need hundreds of thousands of patients and tens of years to detect a difference in mortality between one group and another group. And no, it's very hard to form a company because your investors will come and say, well,
so AI can get you that sample, basically.
So AI can hopefully start deconvoluting these effects so that you don't have to rely on 100 years and not 10 years and 100,000 patients. But that's what's going on in detection.
And treatment?
So treatment is a completely different universe because this is where AI has the maximum impact. And so in treatment, we're using new immunotherapy. We're using new modes of genetically dissecting patients and understanding what drug they're most likely to respond to. And we're using AI in every dimension of guiding treatment so that we can get better and better at treating patients. And that ranges from, more directed guided treatment for the delivery of therapy all the way to, the invention of new drugs, as we just mentioned. So that's what's going on with treatment.
Sid Mukherjee, thanks for the work you're doing.
Pleasure. Thank you for having me.
That's it for today's edition of the GZERO World podcast. Do you like what you heard? Of course you do. Why not make it official? Why don't you rate and review GZERO World? 5 stars, only 5 stars, otherwise don't do it. On Apple, Spotify, or wherever you get your podcasts. Tell your friends.
The GZERO World podcast is brought to you by our lead sponsor, Prologis. Prologis helps businesses across the globe scale their supply chains with an expansive portfolio of logistics real estate and the only end-to-end solutions platform addressing the critical initiatives of global logistics today. Learn more at prologis.com.